You can know precisely, decades in advance, that a disease is coming. Not a risk, a certainty. 40 or more repeats of a single codon and the outcome is written. Most people who carry it choose not to find out. The gene is HTT, and it contains a clock. HTT sits at the very tip of chromosome 4. It encodes a protein called huntingtin, one of the largest proteins in the brain and one of the most important. In a healthy neuron, huntingtin works as a scaffold, assembling the machinery that carries a survival signal, a protein called BDNF, along the long wire of the cortical axon, all the way down to the striatum. The striatal cells at the far end cannot make enough of this signal themselves. They live on what they receive. When huntingtin works, the supply flows. When it breaks, those cells are left waiting.
Physicians had recognized this disease for over 100 years before they found its cause. In 1872, a young doctor named George Huntington described it from Long Island. His father and grandfather had watched the same families pass it down for generations. Chorea, involuntary dance-like movements, dementia, a psychiatric shadow that fell before the body did, and always, never skipping a generation. The search for the gene took another century. In 1983, James Gusella and Nancy Wexler and their colleagues mapped it to chromosome 4, using families from a remote Venezuelan village on Lake Maracaibo, where HD had run through the same kindred for generations, 10 generations deep. The first time a disease had ever been pinned to the genome using DNA markers. But the gene itself took 10 more years to find. In 1993, the Huntington's Disease Collaborative Research Group, almost 60 scientists across 6 institutions, identified it. A stuttered codon, CAG, repeated far too many times in the first exon of a gene they called IT15. Published in Cell, the first trinucleotide repeat expansion disease mapped to its cause.
In a healthy gene, CAG repeats 17 times, maybe 20, maybe 26. That encodes a short stretch of glutamine, a normal part of the protein. Expand it past 40, and everything changes. The protein folds wrong, a sticky, glutamine-rich tail that clings to everything it should not. The cell cannot clear it, and what it clings to, it disrupts. The repeat is also unstable. When a father passes it to a child, the repeat can grow longer still, arriving in the next generation earlier, heavier, harder. Here is what Huntington does every day in a healthy brain. In the cortex, a neuron makes BDNF, brain-derived neurotrophic factor, the signal that keeps striatal cells alive. To deliver it, the cell loads BDNF into a small vesicle, and Huntington assembles the transport machine, a scaffold linking the vesicle to microtubule motor proteins, kinesin and dynein, that walk it down the axon, across the long distance to the striatum below. The striatal neurons receive the BDNF, bind it at their surface, and survive. Huntington also sits in the nucleus and nudges transcription toward more BDNF production. Without that supply, the striatal cells have no backup. Now introduce the expanded polyQ. Fragments of mutant huntingtin, cleaved from the full protein, fold into sticky oligomers. In the nucleus, they trap the transcription factors that normally drive BDNF production, CBP, SP1, holding them in place, away from their targets. The BDNF gene goes quiet. The mutant protein also fails to hold the REST repressor in the cytoplasm, so REST moves into the nucleus and silences BDNF further. At the synapse, the transport scaffold breaks down. BDNF vesicles stall. The striatal cells that depend on that signal begin to starve. But starvation is only half of it. The mutant fragments are toxic in their own right. They clog the cell's cleanup machinery, the systems that should clear misfolded protein backup and fail. And they cripple the mitochondria, the neuron's power plants. Energy production falters. Calcium handling goes wrong. A striatal cell losing its lifeline and its power at once has little left to hold on with. And the cells that fall first are not random.
The D2 medium spiny neurons, the ones wired into the pathway that normally suppresses unwanted movement, lose their grip before any other cell type. When they fail, the circuit that says "don't move" goes dark. What replaces it is chorea, involuntary choreiform movement, the disease's signature. Later, as D1 neurons and cortical cells follow, the movements slow and freeze, and the mind follows.
Huntington's disease moves in one direction. The motor symptoms come first, but the psychiatric shadow often arrives earlier. Depression, irritability, apathy, years before the diagnosis lands. Then the chorea, then the cognitive decline. The disease unfolds across 15 to 20 years.
And it is hereditary, autosomal dominant. One altered copy is enough. So every child of an affected parent carries a 50% chance. The test has existed since 1993. Most people who are at risk choose not to take it. For decades, medicine could only treat the symptom. Tetrabenazine, approved in 2008, quiets the involuntary movements by depleting the dopamine that drives them, but it does not touch the underlying degeneration. Then came the effort to go deeper, to silence the mutant gene itself. The first attempt, an antisense oligonucleotide called tominersen, could lower the level of mutant huntingtin in the spinal fluid. But in 2 large trials spanning 5 years, that biomarker success did not translate into clinical benefit. Tominersen was discontinued in July 2026. Now a different approach is in play. AMT-130 is a one-time treatment, a viral vector injected directly into the brain, carrying a microscopic RNA tool that hijacks the cell's own silencing machinery. That tool finds the Huntington message and marks it for destruction, selectively, inside the neurons where it matters most. At 3 years out, patients who received the high dose showed disease progression that was 75% slower than matched controls. The science has not finished, but the data have moved, and a regulatory application is now in preparation. The first attempt at a disease-modifying therapy is in sight.
150 years, from a doctor on Long Island, to a chromosome, to a codon counted too many times, to a viral tool threading into the brain of the first patients. Marjorie Guthrie founded the society the same year her husband Woody died. She did not live to see this. The families who came after her have kept the search alive. Huntington's Disease Society of America. Research, support, and community since 1967. Find them at hdsa.org. The Gene Channel.